So this is the second video on the fundamentals of biology, and today we will be covering biomolecules. So here are some basic terms that we need to know before we start learning about these biomolecules. The first word is macromolecules, which is another word for biomolecules, which are one of the four main molecules of life. When there is only one building block of one of these macromolecules or biomolecules, it is called a monomer. And when two of these are combined, they produce a dimer. And when there are a lot of monomers combined, it creates a polymer. So how do we even form a dimer or a polymer? Well, this happens through dehydration synthesis, which is when the OH of one group over here combines with the H of another monomer, and they form H2O. And when they leave the system, it will create two monomers that are combined together to form a dimer. And as you can see, water is the byproduct of dehydration synthesis, which means water will leave the system. So how monomers are able to combine to form polymers. So how does hydrolysis work? So it's basically the opposite of dehydration synthesis. It's how polymers will break apart to form the actual monomers. As water enters a polymer, an OH will attach to one molecule, and H will attach to another. And as they enter the system, it will break apart these two monomers and create, or it will break apart the dimer here, or the polymer, and will create two monomers. The first biomolecule is the carbohydrates. So what are the functions of a carbohydrate? Well, they're a source of quick energy, they can store, and they act as sugars. So there are many types of carbohydrates, and they are composed of carbon, hydrogen, and oxygen. Its monomer is a monosaccharide, and related to this, the dimer and the polymer are called the disaccharide and the polysaccharide. So let's just go over some examples of monosaccharides. You can see that some are fructose, glucose, and galactose. And a dimer is when two of these are combined, so there's maltose, lactose, and sucrose. And over here, glycosidic linkage is basically formed through dehydration synthesis and is what we call how they form. So here are some polysaccharides. You can see that they can be categorized into storage polysaccharides or structural polysaccharides. Two of them for storage are starch in plants and glycogen for animals, whereas for structure, there's cellulose in plants and chitin in fungi and the exoskeletons of arthropods. The next kind of biomolecule is the lipid, and what they do is they can serve as part of the cell membrane. They can be longer term energy storage, and they can be used as metabolic fuel. Their functional units are made of carbon, hydrogen, and oxygen, and sometimes phosphorus, as we will see later. And it will have a 3-carbon glycerol, which is this thing on the side, and will also have fatty acid tails. And these bonds between the glycerol and the fatty acid tails are called an ester linkage. And one idea about lipids is that they are nonpolar, which means that it does not mix with other polar objects, such as water. So there are many various types of lipids. The first kind is the triglyceride, which is a glycerol, and then on each of these oxygens, you can see that there's an ester bond and three total fatty acid chains. However, when there are only two of them, it will become it can have 2 fatty acid tails, and on the third part, I guess, there's a phosphate group attached to it, which is where we get the phosphorus, and it is attached to the glycerol. A lipid can be saturated, which means that there's a maximum number of hydrogens and there will be no double bonding formed. So you can see that this is completely straight lines. And since it is all condensed like that, it will form solids at room temperature usually. However, there can be an unsaturated fatty acid, which means there's a kink in the tail, which means there's a double bond because there are not enough hydrogens to sufficiently bond together to make it straight. And since it's missing some hydrogens and there are double bonds, the structure will not be as rigid as a saturated one, so they will be liquid at room temperature. An example of this would be your common vegetable oil. Another kind of lipid is the steroid, and as you can see, it is 4 carbon rings attached together. And a cholesterol is a specific type of steroid, which is found in animal cell membranes. The next kind of macromolecule is the protein, and its basic units are composed of hydrogen, carbon, oxygen, nitrogen, and sometimes sulfur. And we will see why it changes later. So it is composed of a central carbon over here with an amino group, an NH2, a carboxyl group, and a unique side chain. And this unique side chain is unique to 20 different kinds of amino acids. So there are 20 different kinds, and each one has a different side chain, which is why sometimes they can have sulfur inside of it because the R is different. And a polymer of a protein is a polypeptide, and they are formed through peptide bonds. There are a lot of functions for proteins. One of them is to be used as enzymes. They can be used as defense against disease in the immune system. They can store amino acids, other amino acids. They can transport substances. They are used in hormonal activity, and they can be used for structural support. One idea about proteins is their 4 levels of structure, which is increasing in complexity. So the first structure, or the primary structure, is the actual sequence of amino acids, and this can vary between different proteins. The secondary structure are the coils and the folds from the hydrogen bonding among these proteins, which can change the shape and make the protein have different function. Tertiary structure, on the other hand, is the overall shape of the polypeptide, and this can change the protein structure as well. And the final one, or the quaternary structure, is the combining of many polypeptides. So rather than just having one polypeptide, such as in tertiary, there are many polypeptides. And whenever there's a loss of structure of the actual protein, it will denature and it will lose its function. And things that can cause this are a difference in pH, salinity, and temperature. So the final macromolecule is the nucleic acids. So what are the functions of nucleic acids? They are our genetic code, and they make up what are called genes, which are actually used to create polypeptides, or the proteins, which function in our body. Nucleic acids are made of carbon, hydrogen, oxygen, nitrogen, and phosphorus. Its monomer is called a nucleotide, and it is composed of a 5-carbon sugar, a phosphate group, and a nitrogenous base. And they form polymers through a phosphodiester linkage. And there are 2 kinds of nucleic acids, and they are DNA and RNA, and we'll compare them here. So what are the major differences between RNA and DNA? So let's just look at RNA first. So you can see that RNA is single-stranded only, and its nitrogenous bases are C, G, A, and U, which are guanine, cytosine, adenine, and uracil. And its sugar is actually called the ribose sugar, which is why RNA is called ribonucleic acid, because of the ribose sugar, which means that it contains oxygen on the 4th carbon of the nucleotide. And we can compare this to DNA, which is double-stranded and is anti-parallel, which means one strand will be facing the opposite of the other. And its nitrogenous bases are almost the same as RNA. They are C, G, A, and T. So instead of the uracil, it will have thymine. And because DNA is called deoxyribonucleic acid, it will have a deoxyribose sugar, which means there's no oxygen, deoxy, no oxygen on the 4th carbon of the nucleotide. So when we want to pair the nitrogenous bases on the DNA when they're anti-parallel, the adenine and guanine are called purines, and cytosine and thymine are pyrimidines. And the rule is a purine always have to pair with a pyrimidine. So which one pairs with which? Adenine will always pair with thymine, and guanine will always pair with cytosine. I like to remember this because G kind of looks like C, so I guess that can be one way to memorize it. Thanks for watching. This lesson covered dehydration and hydrolysis. It covered the 4 major biomolecules, which are carbohydrates, lipids, proteins, and nucleic acids.